So welcome back, everybody, to another webinar organized by Princeton for everyone worldwide. We're very happy to have Monica de Bolle with us from Peterson Institute. Hi, Monica. So Monica will talk about chronic pandemic, ? And I changed this title slightly to vaccine geopolitics. It will be about policy implications as well. And we're very curious to her take on the vaccine aspects and also on the geopolitical and political implications. Let me give a few opening remarks and then I pass on the floor to Monica. So what's about vaccine geopolitics? Is it like the vaccine diplomacy will come after face mask diplomacy? And first, what is geopolitics? And there are two definitions. One is politics, especially international relations, as influenced by geographical factors. So the Earth, the geographical factors matter a lot. But there's a second definition, which is all about zero-sum thinking. So is it, or do we have a zero-sum thinking? And if you think about vaccines, it's surely not a zero-sum. If you provide more vaccines to more people, that's a positive sum game. And the fixed costs are very high, but then the marginal costs are not so high. But the power which arises from this vaccine might be zero-sum. There will be one country gaining at the expense of other countries. And that's especially true, and I think Monica will talk about this, if we're in a world where we constantly have evolving mutants and various versions of the virus. And it might be a long-lasting game. It might not just be over in 2021, but it might last 10 years or something. And then the question is, who can produce the best technology and how can we distribute the best technology? Is it, the mRNA technology, which is the best technology to adapt to new mutants, like what's produced by BioNTech and Pfizer, Moderna and CureVac, or is Germany and US? And who is the best to distribute it? Is perhaps a vaccine like AstraZeneca or Oxford. The Oxford typically dropped now. I don't know why the name Oxford dropped from the name. And Johnson & Johnson, UK, US again, might be better vaccines to distribute it for, especially for the developing world. And we would like to learn today more about that. So vaccine geopolitics plays a major role. And the question is really, is it, should we have a nationalistic approach or should we have much more multilateral global approach? And if we go back in history, the eradication of smallpox, which killed 2 million people a year in the 60s and earlier, the Soviet Union was very, very instrumental in this. And even though there was the height of the Cold War, there was some agreement with the global partners, essentially, or not partners, enemies, to eradicate smallpox. And that's essentially something we should achieve. Again, in this case, we should not use this for geopolitical reasons, but we should actually harmonize and work together to eradicate that. And so the big global challenge is, of course, you can eradicate the corona crisis in one country, but if it's not eradicated in another country, and if there's a little bit of movement across the borders, the virus will spread. We had an earlier webinar with Stephen Redding, who showed in his model, SIR type models, as soon as there's a little bit of movement across the border, then you haven't controlled the virus. That's one thing. So you cannot really locally contain the virus. The other aspect is if you have partial vaccination, that might be even more dangerous because then the virus can mutate. So it's actually just doing it partially, vaccinate part of the world or part of the population is not the real way to go. So we have to go on a global perspective and on a multilateral approach. So the WHO might play a major role in this. But the question is, has the World Bank or the IMF found its role in this pandemic? What steps they can promote? Or is it more that private organizations, global organizations like the Gates Foundation play a much more important role than the official ones? And why is that the case? It's an interesting question. Why, the private organizations take to some extent the leadership role in this global fight against the corona crisis. The other thing is, there's a geopolitical aspects. There might be a South-South cooperation between India and Brazil. I know that Monica is from Brazil and is probably in Brazil right now, and in Africa. So suppose we are in this long game and it's an annual vaccination for 10 years. Who is building the vaccine structure for Africa? Or it could be that the technology, it seems like the mRNA technology is easiest to employ very fast to new mutants coming from US, Germany, where the leading companies on these technologies are, but the production technology is then provided by India and Brazil. and it's set up in Africa. So it has to have a role where South-South, India, Brazil, and Africa cooperating in order to cover the whole globe. And ideally, I think the World Bank could play a major role in that. So I'm not sure whether they are thinking along these lines, but it would be one way that could actually make a big difference. And there's then also overcome this geopolitical thinking, which might emerge and might be very counterproductive. Now, what else is important when you think about this geopolitical thinking? You might even, as a country, decide, or the leadership of a country might decide, that you might use your own population as guinea pigs, in a sense, in order to have a geopolitical advantage in your foreign policy. So there's some danger on this as well, because you say, I try out some new vaccines very early on, I get a head start on that, and if it turns out well, then I can actually use it for political purposes as well. You might also provide much more data, and Israel is very a good example on that, where you actually provide other healthcare data and combine it with the data. And this whole data story is very important, where Pfizer and BioNTech essentially have an agreement with Israel, delivering a lot of vaccination to Israel, and then these companies can study how effective it is, and it has big benefits for all of the countries around the world. And that's essentially another way we can use it very geopolitically as well. And Israel, I've read just in the FT today, is actually then the excess capacity on vaccination to have, that will give it to certain countries like the Czech Republic, who agreed to that, to put their embassy essentially to Jerusalem rather than in Tel Aviv. So you can see the geopolitical forces playing out already here. Another important component is, of course, when you do something, you have to have some trust in science. So the institutional arrangement in these countries matter a lot. How is it designed? You can develop a vaccine, but if it's not trustworthy, if it's not done by independent scientists, then actually you don't have the geopolitical power. So your own institutional arrangement matters a lot. And what is your arrangement to promote innovation in the private sector and have a private-public partnership within the country? And finally, a big debate is, of course, the international property rights protection. Should we, to what extent should it be given up in order to really get the global coverage? And we will cover all these interesting questions. Hopefully, Monica will talk about them as well. And so I think it's from a geopolitical perspective, it's a very fascinating topic. Vaccine is relatively cheap, and you can have a huge leverage in the international arena. And that's something one has to carefully think about, and how we want to play this out. And in particular, international organizations should play a major role to channel the whole evolvement in the right direction. Now, let me come to Monica's poll questions and the answers you gave us in the opening. So the first question was, how will COVID pandemic end? Will it end when globally the whole global population is sufficiently vaccinated? That's what 56% thought. In advanced countries first, so I shortened the answer at 8%. Never, it will become a chronic like HIV, 35% think. So it's 56, 8, and 35%. So what role will Russia, China, and India play? Will they become global suppliers to the whole world to provide vaccines? That's what 15% think. Supply vaccines mostly to the emerging economies, so they become very influential in the emerging world. 76% think that. Or they don't have any dominant role in vaccine geopolitically or in the vaccine diplomacy. That's 7% think. So the majority thinks that Russia, China, and India together, probably we should have put in Brazil, there too, have the potential to become big players, especially for the emerging market economies. And the third question was, the emerging markets will have widespread fiscal and financial crisis. That's what 32% thinking. Fiscal and financial problems will be mostly local in these emerging markets. That's 53% think this. And they will be fine because there will be low global interest rates, and because of the low global interest rate, you can sustain the high debt burden, debt level, which translates because of the low interest rate to a lower debt burden, interest burden, 15%. So the answers were 32, 53, 15%. So with this summary and a little introduction, I pass on the floor to Monica, who is an economist by training, but has studied epidemiology and the virus intensively in the last few months and the year. And we're looking forward to your introduction or your remarks. Thanks a lot, Monica, for being with us, and the floor is yours.
Thank you, Marcus. It's a pleasure, and it's a pleasure to be here with all of you. I was very interested by your answers, and I think the first one I will address right away. So let me start sharing my screen. And let me say, as Marcus was saying, I am an economist by training. Marcus and I actually were at the LSE, but I have over the past year been studying biomedical sciences intensively at Harvard Medical School, and I've taken a bunch of courses on virology, immunology, and genetics. And a lot of what I'm going to say is going to be more biomedical in nature, but we will get to the geopolitics and the economic policy implications towards the end. So let me start with the chronic pandemic question, which I think most of you don't think is the likeliest scenario. And let me try to convince you of why it probably is. There are three dimensions here, at least three, when we think about why chronic pandemics might be the scenario that we have going forward. On the one hand, there's the nature of the virus. So the virus that causes, as you all know by now, the virus that causes the disease called COVID-19, and COVID, as you all know, stands for Coronavirus Disease 2019. That's why the 19 is there, is SARS-CoV-2. SARS-CoV-2 is, of course, a virus that is closely or very approximately associated genomically with the original SARS-CoV virus. That's the virus that caused the 2002-2003 SARS epidemic. But it is sufficiently different from the SARS-CoV virus, the original one, in several respects that make it, in a sense, more dangerous and harder to control. And that is one of the reasons why we may have a chronic pandemic in the medium term in the cards. The second reason is we have a lot of zoonotic viruses in the world. Actually, they are the most ubiquitous viruses. Zoonotic viruses are those that jump species and have the capacity to jump species. SARS-CoV-2 is one such virus. The flu virus is also a zoonotic virus. And because of human encroachment in the environment, because that has been happening quite intensively, especially in certain parts of the world, and I come from one such part of the world where that's happening, Brazil, where there's a lot of deforestation of the Amazon forest going on, and there are a bunch of reservoirs of zoonotic viruses in the Amazon. The human species might come into contact with these viruses, and they may jump into us. So these risks are on the rise. They have been on the rise for several decades. Epidemiologists and virologists have been warning us of this for several decades. And lo and behold, we got SARS-CoV-2. And we may well be in for other zoonotic viruses showing up. And third is the lack of global preparedness and cooperation that we have seen throughout this pandemic. And that has a number of dimensions. I just list here three of them. One is communication and transparency. We still haven't got our act together in terms of properly communicating and being very transparent about this disease and what it means. This is why there are people who actually think the pandemic is going to end soon, still after being a year into this. Non-pharmaceutical interventions, that's what NPI stand for, and social and political adhesion have been a problem. So adhering to quarantine measures, lockdown measures, using masks, social distancing, that has been, the experience with that has been varied all over the globe. And we do know, because there is evidence, that countries that had more adhesion to these NPIs, to these non-pharmaceutical interventions, were actually better able to control their epidemics. They're not free of epidemics because, of course, the virus travels. But such as it is, these countries have had better responses. Now, a lot of countries have not. The US is one of the countries that has not. Brazil is another, and it's not by chance that those are the two countries where we have the largest number of cases, the largest number of death tolls. And finally, a third point that is of crucial importance is something called genomic surveillance, and that's the capability of being able to track and trace new variants of the virus as we come up to them. And I'll talk about variants in a moment. The next two slides I'll go over very briefly. This is just what COVID-19 looks like. And you have on the one hand viral loads, you have on the other side here, on the other axis, the antibody response to SARS-CoV-2. Antibody response, as most of you probably know, is one of the ways by which our immune systems react. It's not the only one. There are other factors in our immune systems involved in clearing infections, but measuring antibody titers or levels of antibodies, neutralizing antibodies in the blood, is the easiest way to gauge whether or not there's an immune response going on. So this is why we show that. And viral load, of course, is associated with the infectious phase. So here you have a sense of, what happens with the viral load and what happens in several different scenarios, whether you have mild up to moderate illness, severe illness, or critical illness. These are the stages of COVID. You can have, just a disease that clears up on its own, following a usual viral cycle. But this disease does develop into chronic inflammatory conditions and sometimes into hyperinflammatory conditions. And these are precisely the conditions that lead to hospitalization, ARDS, which is acute respiratory distress syndrome, and ultimately ICU admission in some patients. As you can see here, the virus does have a pre-symptomatic phase, which is what makes it dangerous, because during this period, people are asymptomatic and are spreading virus around. Viral replication is extremely fast, and this is for the variants, the earlier variants. The newer variants, viral loads are actually even higher than what we see here on this chart. So people become infectious and they're asymptomatic within this window, and only by, let's say day 8 or 10, or around this point here, around day 8, if you're having a normal viral cycle, is when you're not infectious anymore. So it's a big window. And this is why we have seen the levels of infection that we have seen. This is a chart on testing and the different types of tests that exist. We have focused primarily on PCRs. So these are the tests that we do when we suspect that we have COVID. These are diagnostic tests, so they will tell you for sure whether or not you have COVID. They're excellent from a diagnostic perspective. They're not very good from the perspective of epidemiological control. Why? Because they fail to catch, in large measure, they fail to catch people when they're in this infectious window shown here. PCR testing, because it's a very high sensitivity test, does tend to catch people when they're in the post-infectious phase. The pre-infection phase, because you're asymptomatic, unless you know you've been exposed, you're not going to get a test. So this window here serves very little purpose in terms of preventing, from an epidemiological perspective, preventing spread. So the problem with the PCR from an epidemiological viewpoint is that it's not very good at catching people when they're in the infectious phase, and therefore it becomes much harder to isolate and contact trace. This is why many epidemiologists in the US and other parts of the world have been insisting on what are called rapid tests. These are low analytic sensitivity tests, but they do catch a lot of people in this infectious window here. So they do turn around false negatives. They can turn around false negatives. PCRs are much better at not turning around false negatives. However, they catch people here because they do identify viral loads when they're precisely in the infectious window. So from an epidemiological perspective, where we have failed and what we should learn when we have a new respiratory pandemic at some point is that we need to develop different kinds of tests for different kinds of purposes. Diagnostic is one thing, epidemiological control is something else.
So Monica, Peter Coy has a question to the previous figure. He was surprised that the people get only sick after the viral load is going down. Why is that?
So this is because the way that this virus actually affects us is, and there are many people, so if you look at the orange line here, There are some people for which the viral loads either increase in severe illness and then stabilize. But the main reason why, this is why COVID is so tricky, the main reason why people get severe illness or serious cases of COVID is because of the way the virus interacts with the immune system. So for some people, what happens is that the virus throws the immune response completely out of whack, so that your immune system actually attacks your own bodies, attacks your own tissues, it's a systemic virus, so it's in the blood, it's circulating in the blood. It can settle in any part of your body. We know that the lungs are the chief place where you find SARS-CoV-2, but you can find them in the kidneys, you can find them in the heart, you can find them in your spinal cord, in your spinal fluid. You can basically find it anywhere. And what it does is it completely wreaks havoc with your immune system, so that your immune system actually starts to attack your own cells. And the main reason why we see, for example, hyperinflationary lung conditions in severe COVID is not due to the virus, it's due to the immune response to the virus, which creates hyperinflammation. So that's why, again, it's a really tricky disease. And it also explains why you see these cases of long COVID. So people that, continue having symptoms after they've been cleared of virus, that's because their immune systems don't go back to normal. And this is another thing that we have to be concerned about. This is the SARS-CoV-2 genome. For anybody who remembers high school biology, 30,000 base pairs is the nucleotide pairing, AUGC, because it's an RNA virus. 30,000 base pairs is relatively big for an RNA virus. This is an RNA virus. And here you have the entire genomic structure with the different genes. So SARS-CoV-2 is down here, the virus. It has these 4 structural proteins, the spike protein, which is quite famous. Everybody knows of the spike protein. But apart from that protein, it has a membrane protein, an envelope protein, which is this inside border here. And then it has this envelope protein, which actually envelops the viral RNA. RNA is very degradable material. So the virus has this lipid envelope that basically protects the genetic material. The genome is right here. You can see that there's this thing called ORF, this other thing called ORF-B. These are in genetics called open reading frames. These are genes that encode a lot of different proteins. That's what these ORF1 regions do. The proteins that are encoded here are vital for viral replication, but they're not the structural proteins. The structural proteins are right here. So you have a gene for the spike protein. Amongst the structural proteins, it is the biggest gene, as you can see here. Then you have one for the envelope protein, the membrane protein, and the nucleocapsid protein. This is important because where most of the mutations are happening with the novel virus variant is here in the spike gene, but there have been mutations here as well. And mutations in this region of the genome are associated with viral replication. We just don't know exactly how. Mutations in the spike protein affect how the virus enters our cells. So here's a picture of how the virus enters our cells. Here's the coronavirus. The spike protein binds to this AC2 receptor on our cells. That creates a vesicle, and that's how the virus then enters the cell. Once it does that, it releases RNA, so it releases its genetic material. And what will happen is that this will transform the cell into a viral factory. So at this point, the virus is basically going to hijack all of the genetic mechanisms within the cell to transcribe and translate genetic material. And it will do this in a way that's very distinct from how bacteria reproduce. So bacteria basically are unicellular organisms that just go through mitosis. They divide, and then they have this exponential multiplication process, but they're clones, right? One bacteria is always a clone of another. That's not how a virus behaves. So what a virus does typically is that once it's inside a cell, it starts producing its own proteins, and then it uses the cellular mechanisms to reassemble a bunch of different virions, which are these things here. This is the formation of a mature virion. And the things that exit the cell, so the exocytosis, which is the last part of the viral replication process, is not just one virion, it's usually in the trillions of virions. And no virion is an exact copy of any other virion, because as the virus is going through this entire replication process, mutations are happening. Random mutations are happening in several proteins of the virus. And once it's finally assembled, it will be different from the original. So some of the variants that we have seen show up, they are a result of this viral replication process. But one important thing to note is that once you catch COVID, for whatever, if you are infected, the viruses that you're going to have within you are not exact copies of each other. Okay, and that's part of viral evolution, viruses being what they are. And that's really important to know. The virus replicates either in a host or in a population. When it does, it mutates. And within the same host, as I said, no mature virion is exactly like any other. Mutations are totally random. Many of them will be neutral from the virus's perspective, so they have no consequence. Some of them may be deleterious to the virus, so those copies of the virus, or not copies, but those virions will disappear. And some may be advantageous to the virus. And this is what we are concerned about, because this is what we're seeing more and more of. When a virus finds a new host, a new species, as it is, it undergoes adaptation and evolution. And over time, this leads to what we call greater viral fitness. Fitness is not synonymous with pathogenicity. So if a virus is more adapted to us, for example, that does not mean necessarily that it's more pathogenic or that it causes more serious disease. It just means that it's more adapted to us, period. So if you look at other coronaviruses, the ones that cause a common cold, for example, those viruses are very highly adapted to us, and yet they cause, the common cold, which is really not a big deal. What are mutations? Just for us to get some of these concepts straight, because there's usually a lot of confusion. Mutations are changes in the genome, okay? There are many different types of mutations. I'm not going to go into that. Variants are genomes that differ in sequences by one or by many mutations. That's important because viruses will have variants. That's not the issue. So when you hear, oh, there's a new viral variant, that's totally normal. There are new viral variants within you if you're infected all the time, because that's how they replicate and mutate. The problem are these guys down here, the variants of concern, which I'll talk about in a second. There is a difference between variants and strains. Even virologists are not exactly, this causes a lot of issues within the virologist community, but variants are typically just genomes that differ in sequences, but they don't have any other sort of characteristic, whether strains are variants that have different phenotypes. What does this mean? They've accumulated sufficient mutations that they're either more transmissible, more pathogenic, or more capable of immune evasion. Okay, so there's a bit of a gray area between what variants and strains are. Then are these guys down here, which are really the concerning ones, the variants of concern. So these guys, they have accumulated mutations that may be more advantageous to the virus, and we will call them VOCs for short, okay? Two final concepts. There's a concept called convergent evolution. This is the independent appearance of similar mutations that may increase viral adaptation. And then there's this other concept called selective pressure, which is when external agents or circumstances that affect viral fitness and adaptation show up. They may be positive or negative, okay? There's some examples here of what convergent evolution is. And let me stop here for a second and tell you guys one thing. We currently have, as of 3 weeks ago, we knew we had 3 VOCs circulating in the world. The one that originated in the UK, the one that originated in South Africa, and the one that originated in Manaus, Brazil. As of today, we know of 6 VOCs. So the same 3 that I just mentioned, but an additional one that showed up in Brazil that's now been upgraded to a VOC. Another one that has also been recently upgraded that was identified in California that has now been upgraded to a VOC. And a third one that has just been found in New York, okay, as of a few days ago. The scientific paper on that particular VOC just came out. So as you can see, what's been happening is that since the end of 2020, a whole bunch of these variants of concern started to show up. Earlier on in the pandemic, we thought that this virus mutated relatively slowly, and it ended up accumulating only about 2 relevant mutations per month. But as it turned out, at the end of 2020, a lot of different mutations started appearing in a lot of different variants, and some of them are concerning for the reasons that I'll show you in a moment.
So Monica, why is this the case that they're showing up now and not in the first wave? And is it the case, can you trace back that, you know, the New York one is, the parent is a British one or something? Is there some?
So I will show you guys a chart that shows where, you know, mutations are common or different. But I can tell you one thing. For these 6 VOCs that have shown up, all 6 of them have a mutation called E484K. That's an amino acid change in a very specific portion of the spike protein that has been associated with immune evasion. So for that particular mutation is probably a result of convergent evolution. In other words, the independent appearance of the exact same mutation in different variants of the virus that appeared in different parts of the world. That is suggestive that the virus is adapting and is adapting in ways that make it more advantageous for it to explore certain things that it has learned about us. Now we're talking about the virus as if it were an intelligent being. It's not. These mutations are random, but you know that is how nature operates. The other thing that helps these VOCs show up is this thing called selective pressure. Now, here's where the New York story is interesting because it emulates the Manaus story. Early on, the city of Manaus in Brazil had a terrible experience with COVID. It had a terrible wave. I hate that term, but let's call it wave because that's what people call it. And in the end, studies were done, and it was found that the seroprevalence, that is the presence of antibodies to COVID, was found in 70% of the population of Manaus. New York was never as high as 70%, New York City in particular, but there were portions of New York City, certain parts of New York City, where seroprevalence was also really high. And remember that at that point, people were talking a lot about herd immunity. So, you know, if a wide part of the population catches the virus, then people gain immunity and therefore they're protected from the virus. And this was thought with respect to Manaus. This was thought with respect to certain portions of New York. This was also the policy that the Swedish government decided to follow. However, what we have learned is that the appearance of these new VOCs in Manaus and in New York is probably not by chance. The very fact that these 2 places had these very severe waves of COVID with a lot of people catching it gave the virus the opportunity to explore mutations that actually are able to evade our immune systems. What this means is that, yes, there is the possibility under exploration, it's not been proven, but there is the possibility that this virus can now reinfect people, whereas before we didn't really know. But now we have more and more reports of reinfection. So selective pressure certainly happened in Manaus and in New York. We can't say the same of other countries and other localities where the VOC showed up, but it's something to keep in mind.
So Monica, Elena Marcelino would like to know whether, all this genetic vaccination, can this contribute to more mutations? Is there some connection between being vaccinated and the vaccine and the virus mutating? Or is this not?
That is a very, very good question, and I was going to get to that.
Okay.
The current vaccines that we have, when we have a bunch of different platforms, right? We have the genetic platforms, we have the viral vector platforms, we have the whole virus platforms, and so on. All of these vaccines were designed to protect against disease, specifically designed to protect against developing severe disease. They are now being tested against these new VOCs to find out exactly how well they perform. For the genetic vaccines, so for the mRNA vaccines, we know some, but we don't know everything. What we know is that in vitro, because these experiments have been done in vitro, they have not been done in people, Moderna and Pfizer have exposed vaccinated blood or vaccinated plasma to the South African variant and to the UK variant. And they have found, I have a slide here that actually shows that, but I'll just go ahead and answer the question now, that the antibody titers, so the levels of antibodies that, vaccinated plasma developed when exposed to these variants was lower than when it was exposed to the previous variants in circulation. But because the Pfizer and the Moderna vaccines offer such high levels of protection, in other words, they have these high efficacy numbers, that means that, efficacy has come down, but it's still pretty high. So in principle, there's reason not to be concerned, but that's in principle only because these experiments are in vitro. We will only have a good response to this once we have seen sufficient people having caught these particular variants and having observed how they have responded if they've been vaccinated. So that question is sort of out in the open. And in the process, the virus is continuing to mutate because, as I said, now we have 6. 3 weeks ago, we had 3. So who knows? This is pretty much where we stand. Other vaccine platforms are being tested, some in the same way that Moderna and Pfizer are tested. Others have already done some in vivo testing. So AstraZeneca ran a trial in South Africa with actual people, and it vaccinated those people, and then it sequenced the virus of those who were infected to find out if they were infected with the South African variant. And what they found was that the vaccine was indeed less effective or less efficacious for people who were infected with the South African variant. We have no clue what happens with the P1 and P2 variants. These are the Brazilian ones. And we have no clue what happens with these newer ones, the California and the New York ones that have just shown up. So the studies that exist basically cover the UK variant and the South African variant. This is the spike protein of the virus. So this is the gene right here. And where the mutations that are of concern, where they show up is in this green part right there. This green part of the gene corresponds to this when you look at the protein from a 3D structure. This is the receptor binding domain. So this is the part of the protein that actually attaches to our cells, okay? And the fact that a lot of mutations are accumulating here, it means that the conformation of this thing, which is a 3D structure, is changing, and it might be changing in a way that either makes it easier for the virus to enter our cells or makes the latch of the spike protein much stronger with respect to the receptors on our cells. So this is associated with greater infectiousness, greater transmissibility, and so on. So this is why a lot of these variants of concern are accumulating mutations here, and this is why they are of concern, okay? This is, sorry to bring this up, but this is just so you see 4 of the variants. So there's variant P1, this is Manaus, there's variant P2, this showed up in Rio, there's variant B1351, this is the South African, there's B117, that's the UK. These are the genomes shown here and the mutations that they have. What I want to show you guys is 3 things. this mutation, this mutation, this mutation, and this mutation. So these 4 mutations, they show up in many of these variants. And again, they have shown up independently. Where are they? They're in the receptor binding domain that I've just shown you. So we've got in these VOCs 4 mutations in the receptor binding domain, which do account for possibly or almost surely greater viral fitness that's going on here. Okay, there are also several other mutations in other parts of the gene that we don't know what their significance is. This is just a picture of the spike protein with the mutations. You've just seen that, so let me jump ahead. This is what I mentioned to you guys before. So this is an analysis of plasma from vaccinated people. And the bit to look at is this bit right here. This is the bit that has 3 of the receptor binding domain mutations that I just pointed out to you. And these are the titers of antibodies, so the levels of antibodies. So this is how they used to be with the previous variant. This is what they're now with these mutations. So you can see that they come down. Okay, so this is what I had mentioned before about the vaccines. So there's evidence of convergent evolution. The VOCs may become dominant. The only way we will know is if we have this thing called genomic surveillance. We need to be sequencing these viruses way more than we have been. And immune evasion means that potentially you have a larger pool of susceptible people because of reinfections. What does that mean for vaccines? We've talked a little bit about that before. But let me talk a little bit now about vaccines and global cooperation, because this gets into the geopolitics stuff. So here we have, a chart that basically depicts where different countries are and regions of the world. And as you can see, here, the rich world or the developed world has more doses of vaccines than they actually need. This does not mean that they have more doses right now. Right now, they don't. These are doses that they have contracted. So eventually, they're going to have more doses than they need. But at the moment, what this means is that there is obviously a supply problem for other parts of the world. And since most vaccines are 2-dose vaccines, you can see these countries here in Latin America as a region is right here. These countries here, which simply do not have enough doses. The data for China here is wrong. Okay, ignore China because China does have enough doses. They're producing a bunch of their own vaccines. Vaccines. So there are different types, different platforms, as we've already said. I want you guys to remember the whole inactivated virus because that has to do with one of the questions in our polls. The genetic vaccines and the viral vector vaccines are vaccines that use the exact same bit of the spike protein of the virus to activate our immune systems. Whole inactivated virus vaccines use the entire virus. What does this mean? I'm going to put this in econometric terms, okay? So genetic vaccines and viral vector vaccines, they're great at fit. So if you were to translate this econometrically, they would have really high R2 squares, okay? R2 squares. They'd be great. They're very finely tuned. That's why you see their levels of efficacy being very high. However, because they're very well adjusted to a particular type of spike protein, they lack robustness. What has robustness? The whole inactivated virus vaccine. Why? Because this presents to your immune system the entire virus, not just the portions of it that have mutated. So one thing, we don't yet have the data for that, but what we do from one of the laboratories in China, but we don't for all of them, one thing that many virologists expect is that the whole inactivated virus vaccines will actually fare better when confronted with new VOCs than the very finely tuned platforms. Okay, and that can be understood from sort of a purely econometric rationale. That affects the geopolitics of vaccines in a very big way, because China and India at the moment are the only two countries that produce whole inactivated virus vaccines. Russia is on here because it produces a viral vector vaccine that's being used all over the emerging world. But the fact is that China and India do produce these whole inactivated virus vaccines. When you look at the portfolios of developed countries, what do they have? They have genetic vaccines and they have viral vector vaccines, but they do not have whole inactivated virus vaccines. Could they develop them? At this point, no, because there are already vaccines in use. And in order to develop a whole new vaccine based on a new platform, you have to undergo something that's called a non-inferiority trial. That means you have to prove that that vaccine is superior to every other vaccine in use. And these are very expensive trials, and that use as control groups, not the placebo, but the people who have actually been vaccinated with these other vaccines.
That's, so, Monica, that's a legal constraint. Nobody can change the law.
It is a legal constraint, but it's also in a sense an economic one, because these non-inferiority trials, they're really expensive to run, and they're typically very hard to recruit for when you go by past experience. Now, of course, you know, this is an entirely new scenario, totally unprecedented. So it could be that developed countries start making their own whole inactivated virus vaccines. That could happen. But at the moment, the countries that produce them are China and India. So there is a possibility, back to one of our poll questions, that the whole world at some point actually does become dependent for a period of time, not for a very long period of time, but for a particular period of time on China and India.
So, talking about weeks, months, or years?
It's definitely weeks, because updating the genetic vaccines and the viral vaccines and the viral vector vaccines takes weeks. So we're definitely talking weeks. Whether we're talking longer times than that, it depends on the VOX and how they evolve. So if the VOX become highly complex in terms of the mutations that they accumulate, it could be longer than weeks. Because remember, these genetic vaccines at the moment, they can only use what we call one antigen. So they can only use one portion of the virus. Now, of course, you can change these platforms so that they can use many portions of the virus. And I think this is what we're going to evolve to. Eventually, we're going to have genetic vaccines that use many portions of the virus, but we're not there yet. The platforms that we have don't do this. The only thing that they can do is substitute one bit for another and go with that. And for that reason, if you have very complicated VOX, you might end up being dependent on these whole inactivated virus vaccines, which, you know, puts this perspective of maybe having China and India playing a very big global role, even if limited for some time, for a while.
Can I ask a different question? Can I, if I got vaccinated in some genetic vaccine, can I use another one, a wholly inactivated virus vaccine later on? Can I have
you probably can. And I would say that yes, but we don't know exactly what the time interval should be between one and another. Now, obviously, if the genetic vaccine that you took is completely useless and it's not rendering any kind of immune response in you, you would probably be able to get another vaccine right away. But these are all open questions that we don't have answers for. I mean, at the moment, people are even considering mix and match situations where you get a first shot of an RNA vaccine and you get a second shot of a viral vector vaccine. You know, people are considering these protocols as ways to deal with the VOX. And there are no answers because scientific trials are being run, but there's a whole lot of thinking going on in this field in order to answer those questions. Obviously, you can't have, I'm sort of running out of time because I have to go teach, but obviously there is a very big issue here. When we talk about economic recovery, they're entirely dependent on vaccines. So, you know, in a world where you have different speeds of vaccination happening, obviously economic recovery is not going to happen at the same pace. So countries that are more advanced in terms of vaccines and vaccination campaigns will come out ahead of others. And at the moment, the reality is that is true for developed countries and not for developing countries. So that income gap is going to widen because of the disparity in vaccinations. And of course, that touches on inequality issues, which are valid worldwide. So when you're looking globally, but are also valid locally when you're looking within countries and looking at how vaccine distributions are taking place within countries. So these issues become paramount. This is just to give you an idea here of where vaccine development is. So this is from phase 3, but these are essentially the vaccines in use. So you have the Russian vaccine here, you have the Indian vaccine, you have CureVac, which is a German vaccine that's an mRNA vaccine that's about to come online, AstraZeneca, Oxford. This is China and all of their vaccines, just to give you a sense of how many they've developed. So they've developed 1, 2, 3, 4, 5, 6 out of these.
All different types or all from the same type?
So the CanSino vaccine is a viral vector. So this one is not an inactivated virus, but the Beijing Institute of Biological Product has an inactivated vaccine. Sinovac is an inactivated vaccine. The Wuhan Institute is an inactivated vaccine. And the Chinese Academy of Medical Sciences is an inactivated vaccine. There are only 2 which are not. This is a protein vaccine. This just injects directly the spike protein. And this one is a viral vector. And then you have here in the US, these ones. So you have Pfizer and Moderna, well, Pfizer across sort of the Atlantic. Then you have Novavax and you have Janssen. Janssen is being analyzed tomorrow for emergency use by the FDA. So that's to give you the sort of the vaccine map.
So it seems like the West is much less diversified in terms of which.
Much less, much less.
And that was a strategic choice, or that's just by accident?
Well, what happened was that some of the research into the mRNA vaccines was very well advanced before the pandemic hit, because the mRNA vaccines that were ultimately developed by Moderna and Pfizer, they were being developed for MERS, so for Middle Eastern Respiratory Syndrome. And as it happens, because MERS is also coronavirus, they were developing those vaccines using the spike protein from MERS. And because that research had been going on for several years, they had already identified the conformation of the spike protein, or the forms and the adjuvants for the spike protein that they needed in order to get the vaccine to work. So the really interesting story about the mRNA vaccines, particularly the Moderna vaccine, because this research was going on at the NIH. I'm in DC, so I'm pointing because the NIH is close to where I am. But the Moderna vaccine, which was initially an undertaking entirely by the NIH with MERS, the NIH eventually learned of Moderna and the efforts that they were making. So they joined forces. They developed this MERS vaccine. And when the pandemic hit, when SARS-CoV-2 showed up and the Chinese basically sequenced it, what they did is they picked up the spike protein, used sort of the same conformation that they had used, the adjuvants and so on, for the MERS platform, and they just adapted it to the spike protein from SARS-CoV-2. So that vaccine was actually ready in February of last year. That vaccine was ready a year ago. Then it had to undergo all the preclinical trials and everything else. But that's why these vaccines arrived on the market first, because they were the first to be developed due to MERS, not to SARS-CoV-2, which is a really interesting story. There's actually a really nice video online about the history of the Moderna vaccine, and it's fascinating to watch. So some implications, just to end. Vaccines are going to need to be regularly updated. That's a fact. That's a given, especially in view of how fast this virus is mutating and how many of these VOCs are showing up. What this means, we've already talked a little bit about, it means that this can be a relatively simple process, or it can be a more complicated process, and you might have to use another vaccine in the meantime, like a whole inactivated virus vaccine, for example. We don't know the answer to this question. There is the question of chronic disease burden and what it means for fiscal implications. So about 10 to 20% of people develop long-term consequences out of COVID. They may be debilitating, which means that these people may need to go on welfare and welfare payments. And that has fiscal implications, which are relatively big when you think of the size of the populations involved and how many people have already been affected by COVID. Of course, we have a lot of concerns over debt and deficit burdens, because if we're dealing with a chronic pandemic, we won't be able to be doing the kind of stimulus that we've been doing on an ongoing basis. At some point, we're going to have to find another way of supporting people and supporting economies, which is not what we're currently doing. What we're currently doing is deploying resources for an acute crisis. If we're into chronic crisis mode, then we can't be doing what we're doing now, because every debt ratio in the world is simply going to explode in fiscal deficits as well. Markus, as you know, we have colleagues at the Peterson Institute, which are not very concerned by this. But if we're thinking about emerging markets, this is a reason for concern, a big reason for concern. Inequality will continue to be on the rise because of chronic disease burden, because of the way that COVID affects particular segments of the population, because of a host of other reasons, including policy responses. And this might have political consequences. You know, countries are polarized. In the emerging world, a lot of countries are polarized. You know, politicians that present themselves as populist, nationalist, whatever, may gain ground on top of this. They may not. But there are certainly open political questions to be explored here, because this is very much associated with this chronic pandemic scenario and rising inequality that we're likely to see. We might see higher economic volatility because of intermittent lockdowns, or the need for intermittent lockdowns as vaccines get updated. We may not be able to do sort of a continuous update of vaccines. So as a new Vox shows up somewhere, then that country might have to go into lockdown for a while until you update the vaccine. And, you know, that's going to have consequences for economic volatility. We need to be thinking about the policy implications of that. And finally, you know, there's the central issue of public health and how we think of public health. I mean, as economists, especially as macroeconomists, which is me by training, we tend to leave public health to the side and basically focus all of our attention on exchange rate policies, fiscal policies, and monetary policies. And, you know, public health is just something out there for other people to think about. Well, as it turns out, that can't be the case anymore, especially if you're going to be in a chronically pandemic world. And reshaping economies around public health can actually have good implications, in the sense that public health is actually many sectors, covers many sectors of the economy, has multiplier effects. And I think this is an area, a rich area for research. You know, how does public health, or how does redesigning an economy around public health sectors, how does that generate multipliers for growth? How does that help the fiscal accounts? How does that help actually countries get out of crises? I'll leave these questions with you.
Can I just come back to, I know you have to run soon, to Jim Stochastic question, you know, isn't the one answer then the testing? And why haven't we not allowed certain tests up front? And are these tests working for all the mutations or variants, or, you know, the test is also failing for certain variants as well?
So that's a really good question. Let me try to go back to a slide. My students are waiting for me already, but that's okay. I think I have it here somewhere. No, actually I don't. Never mind. But here's the chart on testing. So many people have been advocating for these rapid tests, and I'm with them. I think we should have those. Rapid tests, what they do is they will measure viral load. A lot of these VOCs are associated with way higher viral loads. So the rapid tests will be great. We need them. We need them urgently. And the faster we get them, the more we're going to be able to isolate people who are infectious. And this is particularly important with the VOCs. We do not have that strategy very much in many countries at the moment. Some European countries have that strategy. Here in the US, we have failed miserably. We do not have rapid tests or enough rapid tests for the population. They're not hard to develop. These are tests that could be sold over the counter in your regular drugstore. You buy a bunch of them, they're cheap, and then you can do them at home. You know, you can do them at home and run them at home. And that will be a big, very, very big and that will have a very big impact on the epidemiology of a chronic pandemic. We need those tests urgently. We can't rely on PCRs alone. And here's the thing about PCRs, and to your question, Marcus. With PCRs, in the case of the UK variant, for example, the only reason why the UK variant was detected in the first place is that it has a deletion. So there's a portion of the spike protein that has simply been erased. And so when their PCR tests that basically amplify a bunch of genes, including the spike protein gene, and what started to happen in the UK is that they found that for a certain portion of the population in the south of England, the PCR tests were not detecting the spike gene. They were detecting the other gene. So they knew the person was infected, but the spike gene had disappeared. And so they thought, well, there are sufficient people with this strange thing going on that we need to investigate and we need to find out why. So that's when they sequenced the virus and they found the UK VOC. So there are instances for which the PCR tests may not be good, depending on how the VOCs evolve. On the other hand, it is possible to design PCRs that detect the specific mutations in the variants of concern. So that's another effort that should be ongoing. It's ongoing in Germany, for example.
So I know you have to go, and but we have to end at a positive note. I got in the chat box many comments and many questions. Some of them are sobering but fascinating. Others are fascinating presentations, very informative but scary. But we have the tradition to end with a positive note.
So here's the positive note. The positive note is we have jumped about 15 years in terms of scientific knowledge and research in 12 months. And that's just that's a conservative estimate, because it's probably way more than that. If we're going into a sort of a chronic pandemic scenario, we are going to have so many more opportunities for development of new scientific technologies that can actually be deployed across the board. So for example, if you take the RNA vaccines alone, the messenger RNA vaccines, yes, they're now being used against an infectious agent, but guess what? They can be used against cancer. So I have studied, because I've been doing these things, I've been studying oncoimmunology and novel therapies for cancer. That's been my latest thing. And as it turns out, the mRNA vaccines offer a huge opportunity for targeting, not as a main treatment, but sort of as a side treatment for a lot of different types of cancer. We may actually have that. We may actually have the possibility of having vaccines for cancer. So when it comes to the sort of technological, scientific advances and medical advances that are coming already with this pandemic, but with its continuation, they're huge. They're huge, and they're going to have a massive impact on everybody, and it'll be a good impact. So
well, I know that BioNTech was working on using mRNA to fight cancer, but they diverted it now to virus fighting. You don't think that's a diversion and hurts cancer patients? You think it boosts? We have at the end, we have an advantage from this COVID crisis.
Yeah, I think we do. Because you see, the tumors arise from within us, and they're basically constituted of mutations. And no tumor is the same, even if it's in the same tissue and it's the same tumor with the same name. Everybody's tumor is different because they accumulate random mutations just like the virus does. So if you're able to genetically sequence the tumor and then get the specific vaccine for the portion of the tumor that's key to fighting it, you can put that on an mRNA platform and inject it into the tumor.
Let's stop at this positive note before we get some other more sober notes. And I thank you a lot. It was very illuminating, very fascinating. And keep up the good work. I'm following you, and I hope many others will too. And hope to see you soon, at least virtually.
Thanks
again.
Thank
Bye-bye.
you very much.